Hello everybody, welcome to Vegas. My name is Karla Garcia and I am a third-year undergraduate at Nevada State University. I'm working in Dr. Kelly Tseng's lab at the University of Nevada, Las Vegas. Here we use Xenopus laevis, commonly known as the African clawed frog, to study tissue regeneration. Did you know that Xenopus were once used as highly dependable and mainstream pregnancy test? This test took advantage of the fact that Xenopus has a similar hormone response as humans. From the 1930s to 60s, the Hogbin test took the urine of a woman and injected it into a female frog. If the woman was pregnant, the frog would lay eggs the next day. Xenopus is an excellent model to use for research as it is a well-studied, hardy animal that can be induced to lay eggs multiple times a year, facilitating the study and use of frog embryos at various stages. It is also more closely related to us than other models. Xenopus is also highly regenerative and can regrow many body parts. In the Tseng lab, we study how Xenopus regenerate their organs, including appendages and eyes. My lab showed that Xenopus embryo eyes regrow successfully after ablation. We are interested in comparing eye development and eye regrowth to understand the similarities and differences between the two mechanisms. My project focuses on characterizing and comparing lens development and regrowth. The lens is a clear structure in the eye that sits behind the pupil. It helps to transmit and focus light on the retina. The lens is made up of multiple layers, like an onion. These layers are made up of fiber cells. These fiber cells do not have a nucleus, but ironically, this central core of the lens is known as the lens nucleus. The middle layer is the cortex, and the outer layer is known as the lens capsule, followed by the most outer layer called the lens epithelium. In contrast to the cells in the inner layers of the lens, each of these epithelial cells contains a nucleus. In my project, I am examining frog lenses during development by identifying the position of the lens nucleus in Xenopus. This is a collaboration with Dr. Irene Varnsova at the University of Auckland, who studies lens development but in zebrafish. Together, we will compare lens development in Xenopus with that in zebrafish. Once we have this information, I will then examine regrown frog lenses at the same developmental stages to see whether they are the same or different. This study will enable us to compare the lens structure between Xenopus laevis, both in developmental and regrowth, and zebrafish. This project will help us to better understand if lens development and regrowth in Xenopus are similar or different. And on a bigger scale, to see whether Xenopus and zebrafish share the same mechanism for lens development. Figuring out how frogs make their lens will help us better understand the basic process of eye formation and the development of vision in aquatic animals.